The nerve running from your lower spine to your big toe is a single cell. Not a chain of cells passing a signal along. One cell, one continuous piece of living material, up to a meter long and a tall adult. That's an extraordinary shape for a cell to be. And it creates a problem that I don't think most people have ever considered. Let me set it up. Mitochondria are not manufactured everywhere in a cell. They're built near the nucleus, in the main cell body, where the machinery for producing their components sits. Now think about the geometry of that neuron. The cell body is near your spine. The far end, where it connects to muscle or reports sensation, is in your foot. That far end has serious energy requirements. Firing a nerve impulse means pumping ions back across the membrane afterwards to reset, and ion pumping is one of the most expensive things a cell does. Neurons are among the most energy-hungry cells in your body, but there's no factory down there. Everything has to come from the cell body. So every single mitochondrion at the far end of that axon was physically transported the entire distance. Here's how. Running along the inside of the axon are microtubules. Think of them as rails. They're polarized, meaning they have a distinct orientation, and in an axon they're arranged with one particular end pointing away from the cell body. Two families of motor proteins run on those rails. Kinesin travels toward the far end. Dynein travels back toward the cell body. Because the rails are oriented consistently, that means kinesin carries cargo outward, and dynein brings it home. And I want to be literal about the word travel. These motors have two structures that function as feet. They move by alternately releasing one and placing it ahead of the other, along the microtubule, each step powered by ATP. They walk. Now, the motors don't grab the mitochondrion directly. There's a coupling system. A protein called Miro sits in the outer mitochondrial membrane, with part of it sticking out into the cell. Another protein, called Milton or tract depending on the organism, binds to Miro on one side and to the motor on the other. So the arrangement is mitochondrion, anchor protein, adapter protein, motor, rail, a tow hitch and a coupling, and then the engine. Alright. Now the question I find most interesting. Once a mitochondrion is being carried down a meter-long axon, how does it know where to stop? Because that matters enormously. Energy demand along that axon is not uniform. Some regions are working hard, others are quiet. You want your power supply parked at the busy places. The answer is calcium. When a neuron fires an action potential, calcium enters the cell. And Miro, the anchor protein sitting on the mitochondrion, has calcium binding sites on it. When calcium rises, that sensing pathway suppresses mitochondrial movement. The transport complex is disrupted, and the mitochondrion stops. Take a moment with that, because it's beautifully economical. The mitochondrion isn't being directed to a location. Nothing is calculating where it should go. It simply keeps moving until it enters a region where the neuron is electrically active, and the activity itself stops it. Wherever the cell is working hardest is where the power supply comes to a halt. Demand parks the truck. Then there's a second stage. A protein called syntaphyllin acts as what researchers call a static anchor. It's specific to axons, and it docks mitochondria to the microtubule scaffolding, physically bolting them in place. It works by competing with a transport adapter for binding to the motor, and by inhibiting the motor's activity directly. So it's not just holding on. It's disabling the engine, and you can test that. Delete the syntaphyllin gene in mice, and the proportion of mobile axonal mitochondria increases robustly. Without the anchor, they keep drifting. In a mature neuron, the balance sits at roughly 70% of mitochondria stationary, and 20 to 30% moving at any given moment. Most of the fleet is parked. A minority is in transit, replacing and redistributing. There's a connection here to something I covered a few weeks ago. In muscle, I talked about calcium as the signal that triggers contraction and simultaneously switches on energy production inside mitochondria. Calcium says go. Here, in a neuron, calcium says stop. Same molecule, opposite instruction, different tissue, which tells you something about how signaling actually works. There's no universal meaning to a messenger. The meaning is entirely in what machinery is listening. Now, let me be honest about the state of this field, because it's less settled than I've made it sound. Some of the findings conflict. The picture I described, where Miro is essential for getting mitochondria out into the axon, comes largely from work in fruit flies, where removing it causes dramatic loss of axonal mitochondria. But in cultured neurons from mice lacking Miro, the amount of mitochondria in the axon looks normal. The distribution in the dendrites is disturbed, but the axonal supply isn't. And a more recent study suggested Miro isn't required for that transport in another mammalian cell type either. The researchers describing this state it plainly. These conflicting data suggest that our understanding of mitochondrial transport in living animals is incomplete. So the core is solid. Motors carry mitochondria along microtubules, Calcium regulates where they stop, syntaphyllin anchors them. The specific roles of individual adapter proteins are still being worked out, and results differ between species and preparations. I'd rather give you that than a tidy version. What's the takeaway? There isn't a lever here. Nothing to do, nothing to change. This is cell biology running whether you attend to it or not. What I'd offer is the image itself. Right now, in the nerve running down your leg, there are mitochondria being walked along microtubule rails by proteins taking individual steps. Some are heading out. Some are coming back for recycling. Most are parked at busy junctions, held in place by an anchor protein, waiting. That's happening in every long neuron in your body, continuously, and it has been since before you were born. Now, where my own recommendation sits, and it's narrow as always. The motor's doing that walking run on ATP. So does the ion pumping at the far end that makes the whole delivery necessary. That ATP comes from the electron transport chain, and CoQ10 is one of the carriers within it. Levels tend to decline as we age. That's a claim about energy production. Nothing here is about supplements. Nobody has studied one against axonal transport, and I'm not implying a connection. The link in the description below goes to a free 5-item morning checklist. Mitolin's on that page as well, and it's an affiliate link. I earn a commission at no extra cost to you. And if you want more of this, subscribe. This video is educational only and it's not medical advice.